my dear students, I hope all of you are doing very well. You are every day you are learning, and we are actually continuing our this journey of pharmaceutical biotechnology. This is the 28th lecture in our particular this subject and second lecture in unit 4. My name is Abhijit Devnath. I am working as an assistant professor at NIT Pharmacy Institute, Greater Noida. So, today I am going to tell you something very interesting thing. I hope you have enjoyed my previous lecture where I have told you about ELISA. Today I am going to tell you about human blotting techniques, how you can use western blotting, northern blotting, southern blotting to identify the presence of certain DNA, protein or RNA in a sample. So, today we are going to learn about, at first I will tell you some basic terms, which terms will be very much important for us to understand the blotting techniques. Then I am going to tell you about the introduction of blotting techniques, then what are the applications where we are actually using these blotting techniques, and one by one individually southern, northern and western blotting techniques I am going to tell you. You might be wondering that south, north, west, where is east? I am going to give you the answer. You may just continue listening the lecture, I am going to give you the answer, where is east. Blotting techniques are actually given in the name of some direction, there are some reason, that is also a mystery. I am going to also tell you in this lecture why we have given the name of the various blotting technique in the name of direction. Okay. So, enjoy. So, let me start with the basic terms. Electrophoresis. So, what is electrophoresis, sir? So, it is an electrokinetic process in which separates charged particle in a fluid using field of electric charge. See, it separates charged particle in a field using field of electric charge. Here you can see, here is a typical picture of electrophoresis activity is happening. Here you can see electrode negative, and obviously in the other side it will be positive, okay, electrode. So, it is a electric field we have actually generated. Here we have buffer solution, and here we have given some sample. In that sample there are chances that some charged particles are actually present. So, those charged particles it will be get separated, you can see in this is sample number 1, this is sample number 2, 3, 4 and 5. So, within all those samples you can see for example, you can see this, here is something gel, okay, gel is also present, these are our samples. So, you can see in these two areas it is separated in a uniform manner. It means, in all the samples this substance are actually present. So, basically it is a process by using which process we can separate charged particle in a field using field of electric charge. The procedure differs in some way, but all needs to a source of electric charge and a support of medium and buffer solution. So, it is used in laboratory for separation of the molecules based on size, density and purity. So, what are the molecules it will separate it? It will separate it based on its size. Large particle will be remain here, small particle will be come here, okay, and also its density and purity, based on its density and purity it will separate. And if I tell you more, so actually we are actually applying an electric field to the molecules, as they are electric charge themselves, it results force acting upon them. So, if a molecule actually present is here, so based on its charge, obviously you are aware that if here is some negative molecule is present, here is some positive molecule present. So, what will happen? Negative will be go with the negative and positive will be positive. So, ultimately it will be that is there will be a run of molecules like this. Here gel is present, and you can see the separation, you have seen the separation. So, the greater the charge of the molecule, the greater the force applied by the electric field, and therefore, the further through the support of the medium the molecules will move relative to its mass. If I tell you more, see that is I give you some more clear picture. Here is a very good picture. So, you can see these are the few that is the sample. Okay. So, this lot containing DNA sample actually is present here, DNA sample. So, electrode was actually present positive and negative. So, ultimately when you have applied in gel solution, when you applied this positive negative charge, so ultimately the molecule run was actually happen. Run means based on its charge and mass, it will move from here to here. Okay. High density molecule will be remain there. So, those molecules having a very that is a short or small particle, this will be able to come up to here, and you can see big particles are here and small molecules actually come here, also based on its charge, also based on its density. So, this is actually electrophoresis. By using this technique you can separate the molecules based on its charge. And also actually we are actually using here gel, we are actually using here this thing, this area whatever you can see yellow color, all the things are actually gel, that is why it is called as a gel electrophoresis. So, these gel electrophoresis techniques can be used in separating in DNA fingerprinting, DNA analysis, protein and antibody interaction, testing antibodies and testing vaccines also, these things can be used. Now, let me show you some picture. You can see here is the various kinds of antigen like anti-human antigen and anti-IgG, IgA, IgM, all are antigen, okay, kappa and lambda, how they are actually moving based on their charge. Here is another picture, you can see you will be after completion of your the entire activity, entire that is experiment, you will be getting such kind of band, such kind of results. By using this bands, you will be able to analyze, you will be able to tell what is its molecular weight. Okay. So, you can see these are the few other pictures. So, ultimately these things you are going to get after completion of your work. Now, come to the Immuno-Blotting technique. I hope all the basic terms are very much clear to you all. I hope it is very much clear to you all the basic terms. Now, come to Immuno-Blotting technique. So, in this Immuno-Blotting technique, use antibodies to bind target protein among a number of unrelated protein species. Okay. In this technique actually uses the antibodies to identify target protein among a number of unrelated protein species. They involve identification of protein target via antigen antibody or protein ligand species reaction. Blots are techniques for transferring DNA, RNA and protein into a carrier. So, they can separate it and often follows the use of the gel electrophoresis. For example, like this southern blot, it is used for transferring the DNA. Northern blot, it is used for the transfer of the RNA. Western blot, we actually do these kinds of blotting technique for protein, and here is the eastern blot also for protein. You remember at the beginning of my lecture I have told you where is east, here is the answer, here I am solving the mystery. Eastern blotting actually used for the protein and post translation modification including addition of lipids, also this thing called phosphate and glycoconjugates. So, eastern blots are actually not so much popular, it is also not a part of your syllabus, that is why I have skipped, I am not going to tell you too much about the eastern blotting, but for your knowledge, for your curiosity, I thought I should tell you that where or what kind of, for what kind of activity we are actually using eastern blotting. Okay. So, I hope it is very much clear to you all. You should always remember that southern blotting is used for the DNA, northern for RNA and western for protein, western and eastern both the things are for the protein, especially for post translation modification. Now, principle, what is the principle? Principle is very simple. After learning the basic terms, I hope it is very much clear to you all what might be the principle. Principle is the mixture of the molecules will be separated, the molecules are immobilized in a matrix. The probe will be added, one probe will be added to the matrix to bind the molecule. Any unbound probe are actually then removed, and this place where the probe is actually connected corresponds to the location of the immobilized target of the molecule. This is the basic principle of Immuno-Blotting technique. Now, come to here. So, these are the 4 types, these are the 4 different types of Immuno-Blotting technique. So, you can see western blotting we are doing for to detect the protein, southern blotting we are doing to detect the DNA, northern blotting we are actually doing to detect the RNA, and eastern blotting we are also doing again for protein. So, for protein eastern and again western, okay. And for DNA southern, and for RNA we are actually doing northern. Okay, you should remember this picture, otherwise you will be confused in examination that western blotting is used to detect for what, you know. So, you should remember. Now, where electrophoresis is actually lying, or what are the differences between these basic techniques. So, target molecule for southern DNA, northern RNA and western for protein. So, you have the target DNA, you have target molecule, you have certain DNA, you have some DNA sample or you have some RNA sample or you have some protein sample. You have taken that sample, you are trying to see that or you are trying to separate the various protein complexes, various protein mixture based on their that is a mass, molecular mass. Okay. So, in this regard, in this process, if your protein you are going to do Western blot, if it is RNA northern, if it is DNA then southern. First of all, you have to prepare the sample, you have to prepare the sample. So, DNA extraction and enzyme digestion, in these cases you have to separate the RNA, that is RNA isolation. Here you have to do the protein extraction, and then the most important and most interesting thing, electrophoresis. I hope now it is very much clear to you all why I have told you in detail about the electrophoresis. Here electrophoresis is come to the picture. You can see all the cases, in all the kinds of blotting technique, electrophoresis will be used. But yes, after that some membrane material, in this case DNA you are going to use the nylon, for RNA again you can use nylon, but here it is big molecule, that is why I am going to use the this thing called nitrocellulose or PVBD. What are the probes? What kind of probe you can use? Nucleic acid probe with sequence homologous to the target, nucleic acid probe, ITGC. In this case it is RNA. So, what will be the probe? The RNA, DNA or oligonucleotide. It is protein, so obviously primary antibody we can use. Now, probe level. In probe level, how we can quantify, how we can that is confirm. So, radio level or enzyme probe level you can use, and in this case of RNA or northern blot, the radio level again enzyme, but in this case Western blotting only enzyme you can use. Now, what are the detection method? What are the detection method? X-ray film or chemiluminescence. In this case again X-ray film or chemiluminescence, and in this case film coated CCD film or that is a cold CCD camera or LED or in this thing called infrared imaging system can be used. So, I hope it is very much clear to you all that how these things are actually happening. So, you have taken the sample, any of these things, you have to prepare the sample, you have to do electrophoresis in all the cases, then you have to use some membrane material, in those cases nylon, in this case this thing called nitrocellulose PDPF. Probe, you can use the same probe for both this case DNA and RNA, here you have to use the primary antibody, and probe level again same, radio level and enzyme, but in this case only enzyme, and at the end at the last detection level X-ray film or chemiluminescence same in both this case, and in this case only film or cold CCD camera etc., etc. Where we can use this kinds of technique? We can use this kinds of technique obviously to detect the presence of the DNA, RNA etc., etc. So, southern blot allowed to investigator to determine the molecular weight of the restriction fragment or measure the relative amount of the different sample. Southern was also used to detect the presence of a particular DNA and analyze the genetic pattern in which appearance of the person's DNA. Okay. So, let me tell you one by one about all the various techniques, but yes, I will not tell you about the Eastern blotting, because this is not a part of our syllabus, I am going to skip, but remaining 3 I am going to tell you. Let me first start with the southern blotting. So, southern blotting, this is used to in molecular biology to detection of the DNA sequence in a DNA sample. It is very much clear, because lots of times, several times I have told you. And this technique southern blotting combines transfer of electrophoresis separated DNA fragment to filtered membrane and subsequent fragment detection by probe hybridization. The method is named after British biologist Edwin Southern. See the name, it is his surname Edwin Southern. I am going to show you his picture in my upcoming slide. So, that is why to respect him, because ultimately in 1975 he actually worked on and he ultimately given this technique. To show a great respect to him, all the other technique given in the form of direction. Here southern for DNA, when it will be northern RNA, protein eastern, post translation modification of protein western, okay, eastern and western. Now, how you will do or what will the process, what are the steps? First, restriction digest DNA, that is restriction in endonuclease, digest DNA will be there. Then you have to do electrophoresis. Obviously, you have seen about electrophoresis. Then you have to transfer, Southern transfer, and after that there will be some paper, etc., gel, etc. thing, and then radioactive probe will be there. Radioactive probe. After that, will be washed. What are the remaining things? It will be get out, and then probe hybridized to restriction fragment. It will be done. Then X-ray film. X-ray film will be produced. Ultimately, you will be getting some developed X-ray film. Here you will be getting the band, and this will determine the size and restriction fragment that hybridize the probe. Okay. So, you can see how the things are actually working. First you have collected the sample, you have done electrophoresis, and then you have the radioactive probe, and ultimately you got the X-ray film. Based on that, you can tell that how much or what is the that is a size of restriction fragment or that hybridize the probe. So, other blotting methods, for example, like Western, Northern and Eastern, that also employ the similar technique, but using the RNA or protein or map later, but actually we actually given the name Southern in the respect of the Edwin Southern on his name. Okay, as the level of that is a Epocynamus, Southern is actually capitalized as it convention of the proper nouns. The names of other blotting techniques method also follow the same convention. Okay, you remember these things. So, this is the ultimate thing whatever you got, and he is our great scientist Edwin Southern. To show our great respect to this great person, we have given the name called Southern blot. His name is Edwin Southern. 1975 he has actually developed this technique. Now, this is the entire process whatever I have told. I have again tried to give you much more clear picture, so that you can understand. You can take screenshot, or if you want to write in your examination, you can take these things. So, ultimately after doing electrophoresis, your everything will be get separated, and then you have to do the DNA fragments that are bloated, whatever will happen, and then you have to do the filter by using radioactive probe, and then ultimately that filter probe you will be putting this under X-ray film. From that you will be getting the results. So, application obviously we actually using for DNA based activity. It is also used for the homology based cloning, basis of the amino acid sequence, and also oligonucleotides are designed so that they are similar to the target sequence. The oligonucleotides are chemically synthesized, radio labeled, are also used to screen a DNA library, other things. Sequences that hybridized within the hybridization probe are further analyzed, for example, obtain the full length sequence. Okay. Southern blotting also can be used to identify methylated sites, also particularly useful for restriction endonuclease. Okay. So, this is something about the Southern blotting. We have covered this part DNA. Now, come to the other thing for RNA. Let me talk about the RNA, the Northern blotting. Now, Northern blotting actually used to determine or identity, size and abundance of specific RNA in a sample. This Northern blot protect the begin with the RNA isolation and separation techniques may vary based on the RNA size. So, large RNAs are actually separated by electrophoresis on a formaldehyde agarose gel, or this thing called glycol agarose gel, which prevents normal base preparing and maintains RNA in a denatured state. Small RNA are actually separated on denatured or polyacrylamide gel. So, you can see this is the area where they are actually working. So, RNA actually is then transferred from the gel to nylon membrane is then incubated with a radioactive or this thing called non-isotopically labeled RNA or sometimes DNA or oligonucleotide probe, and then unhybridized probe actually removed by washing in a buffer. You can see this is the process. Here you have the entire sample, from that you have done the RNA extraction. So, you have done electrophoresis, and then you have ultimately got these things. From that electrophoresis, whatever the that is a plate actually present here, from that you actually you actually getting separated these things. So, radio label probes are actually visualized with the X-ray film, and enzymatically labeled probes are actually visualized with the chemiluminescence. Okay. So, Northern blot technique was actually developed 1997. In 1975, Edwin Southern, he developed the Southern blotting. After 2 years in 1997, Northern blotting was actually developed by whom? By James Alwyn, David Camp and George Streck in Stanford University. Okay. So, you can see these are the various Southern blotting picture of brain, liver, brain cells and other cells. So, what are the RNA actually present, what are the RNA actually responsible for that, you can see the picture. This is the process, you have the RNA, you have done electrophoresis, separated the RNA, then you have transferred the RNA from gel to membrane, so that you can visualize the RNA. See. So, basically we can separate, identify and quantify the amount of RNA present in a certain sample. Application obviously for RNA we are actually using Northern. So, it allow one to observe particular genes expression pattern between the tissues, organ, development stages, environmental stress level, pathogenic infection over the course of treatment. This techniques has been used to show overexpression of oncogene, down regulation of tumor suppression gene in cancer cells when compared to normal tissues. Okay. Also, we can use if upregulated genes is actually observed, an abundance of mRNA on the Northern blot sample can be then sequenced to determine the gene known as the researcher, if it is a novel finding. Okay. To get a novel gene, you can also use this technique. In other cases, you can also use the expression pattern obtained under given condition can provide insights into the function of the gene. Since RNA is a first separated by size, if only one probe type is actually used, variance in the level of each band on the membrane can provide insight into the size of the product, suggesting alternative splicing, alternative splicing product of the same gene or repeated sequence of motif. Can you imagine how much great this technique, what scenario we can use. So, these are the few applications of Northern blotting. Now, let me tell you about the Western blotting, because ultimately we actually use this to detect protein or to separate protein. So, let me tell you about the Western blotting. This is very simple. In this case, obviously we are actually using some enzyme, antibody enzyme, all the reactions things actually happening. So, Western blot is actually used to determine the identity, size and abundance of the particular protein in a given sample. So, Western blot protocol begins with the sample lysis preparation from the tissue or cell culture and separate the polyacrylamide via gel electrophoresis. Okay. These separated proteins are then transferred to a nitrocellulose or polyvinyl this thing called difluoride membrane, PVDF membrane. So, the membranes, whatever the membranes actually are getting, this actually incubated with a block agent to prevent the non-specific binding. See, there are lots of proteins might be present, there is a protein of your interest. So, more non-specific binding you have to avoid. In this case, you are actually incubate with a blocking agents to prevent the non-specific binding, followed by incubation with a primary antibody to bind a protein of interest. There are two detection method, direct and indirect. So, in direct detection method, it relies on the labeled primary antibody, and indirect detection method, a primary antibody is actually directed against the target protein and a second antibody directed against the immunoglobulin class or subclass of the primary antibody species. Okay. There are two different methods, direct, you can either go with the direct detection method or you can go with the indirect detection method. Okay. Now, visualization, after doing all those things you have to visualize now. So, in visualization method actually includes the colorimetric assay, in which color precipitator actually produced, chemiluminescence or fluorescence can be used. Here is the entire protocol. So, here is the antigen sample, you have separated the protein by using the gel electrophoresis, then this is the blotting tank where proteins are actually transferred to nitrocellulose sheet, and here you have added the label protein. You are aware why actually we have added the label protein, just now I have told. Then after that what you are doing to do? You are doing the autoradiography, and then you have got the antigen band, and through that you can tell the you can get the visualized antigen band. So, the Western blot is actually extensively used in biochemistry for the qualitative detection of single protein and protein modification. such as post translation modification. So, it is also used in generally to identify the presence of specific single protein with a complex mixture of the protein, and a semi quantitative estimation of a protein can be derived from the size and color intensity. And in addition, applying the dilution series of a purified protein known as the concentration can be used, allow more precise estimation of the protein concentration. Okay. So, this is the membrane, here is the primary antibody and here is the labeled secondary antibody. See, through that you can confirm that yes, is a present. Where we are actually using this thing? We are actually using in obviously in protein, for protein that is a size, etcetera, etcetera. This also used in the medical diagnostic like HIV or BSC test, and also used in definitive test, okay, various diseases, and we can also use in the this thing called tularemia, an evaluation of the Western blot ability to detect the antibody presence F tularemia revealed its sensitivity up to 100%. Okay. I was telling you at the beginning of the lecture that we can use this technique for various kinds of that is a microbial detection kit. We are actually doing such things. You can see these are microbes. Some form of Lyme disease testing can be also employed, Western blotting, and Western blot can also be used to the confirmatory test for hepatitis B infection, okay, or HERVS type 2 infection. So, today we have learned about the basic terms which are actually related to immune blotting techniques. We have also seen what is immune blotting techniques, application, where you can use. Then I have told you southern blotting, northern blotting and Western blotting. So, that is it for my side for today. Tomorrow we are going to learn something more. Tomorrow I am going to tell you about the prokaryotes and eukaryotes, what are the difference between them in genetic level, in genomics level. Also how these things are actually playing a big role in microbial or our biotechnological activity. Okay. So, these things are our next tomorrow's discussion, these topics. So, thank you all of you for joining me for today's lecture. I hope all of you have enjoyed this lecture, and I hope this lecture will help you in your future research also, because these techniques, immune blotting techniques, these are the base of our biotechnology. So, thank you all of you for joining. God bless you all. We may meet in our next lecture. Thank you.